Hello, grade 11 students. In this video, we're going to continue our lesson about genetics. In this video, we're going to review together the steps of the factorial analysis. So the first step, we have to write the phenotype of the individuals or the parents. Second step, we have to write the genotype of the individuals. Third step, we have to write the gametes. And the fourth step is going to be the table of cross, and the last step, which is step number 5, table of results. Let's move to talk about the case of codominance. As we said before, the codominance is the co-expression of both alleles, where both alleles they are dominant and both of them they are expressed. In some cases, both alleles they are independently expressed. Example about that of the allele A and B of the blood group. So this alleles A and B in the blood group that determines the blood group AB of the ABO system. Father and mother they have blood groups A and B respectively. So let's move now to the first case where the parents are pure. So let's start first. We have to write the phenotype of the parents. So phenotypes, we have male, mating cross, we have the female. We said that the father has blood group A. So blood group A, you can put it here between these parentheses, this means the phenotype. And here blood group B, the female. Moving for the genotype of the parents, so male and female. So the genotype of the parents they are pure, meaning that they are homozygous. So they have the genotype blood group A, AA, and the blood group B, it's BB. Moving now to the gametes, the symbol gamma. So gametes, here we have AA. So the possibility to have A, it's 100%. So this is A, 100%. And here to have B, also it's 100%. Moving to the fourth step, which is the table of cross. Table of cross, going to draw the table of cross. Male and female. First of all, you have to divide it, male and female. The male has gamete A 100%, and here we have B 100%, and they will give the genotype AB 100%. Moving to the table of results. So the table of results. So the genotype will code for the phenotype. So you write genotype and phenotype. So the genotype is AB 100%. So 100% we have individuals of blood group AB. Here both alleles A and B they are expressed. And as we know in the ABO system, the alleles A and B they are dominant, while the allele O is the recessive one. So both alleles here A and B they are expressed. They are co-expressed. So this is the case of codominance where the parents are pure in the case of blood group. While if we have moved to the second case where the parents are hybrid. So let's move. Hybrid for the blood group A, what will be the genotype? And hybrid for the blood group B, what will be the genotype? To start first, we have to move the steps of the factorial analysis as we did before in the first case where the parents are pure. So let's start first with the phenotype. So phenotype, male cross female. We said that the father has blood group A, so blood group A, and here we have blood group B. Moving to the second step, the genotype. So the genotype for the male And for the female, we said that they are hybrid, meaning that they are heterozygous. So first of all, they have A, and here they have B. So they have A and they have B. So both of them, they are present. They are hybrid. In order to be blood group A and you are hybrid, you can be AB. Since it's A and B, it will be blood group AB. So it should be with the one which is recessive. So it should be with the O. And B, in order to be hybrid, should be with the O. So B and O. So that's why if some parents, they are blood group A and blood group B, and they gave birth to, for example, children with blood group O, it's not weird. It can happen, and we will know how. Moving to the gametes of symbol gamma. So here in the gametes, we have both gametes. We have A, which represent 50%, and O, which represent also 50%. And here B, which represent, and the female gamete B represent 50%, and the O represents 50%. Moving to the table of cross. Table of cross, as you know, you have to draw the table and you have to divide it into 4 boxes. So here we can divide it here, and here we have male and female. You have to pay attention here, please. The male, it has is A 50% and O 50%. The female has B as 50% and O as 50%. So to start first, we have A and B, so it's AB. 50 times 50 divided 100, so it's 25%. While O times B, it's OB. It's not OB, we start with the dominant always, so it's BO, 25%. While O times A crossed with A, so it's AO, 25%. And the last one, it's OO, which is 25%. Moving to the table of results. Table of results. In this table of results, we'll have different results, as we will see. So genotype will code for the phenotype. So first of all, we have AB, 25%. This will code for what? 25% individuals having blood group AB. And we have BO, 25%. And since B is dominant over the O, which is recessive, so we have 25% individuals having blood group B. AO, we don't have other than this AO, 25%, meaning that 25% having blood group A, and OO, 25%, meaning that we have 25% having blood group O. So here we have 4 different results, 4 different blood groups. That's why if individuals have blood group A is crossed with individual have blood group B, they will have possibility to have blood group AB, blood group B, blood group A, and blood group O. So if they inherit, for example, the A from the father and the B from the mother, it will be AB. If they inherit the A from the father and the O, it will be AO. This is the case here. And if they inherit, for example, the B from the mother and the A, it will be AB. B and the O, it will be B, and O, it will be O, the blood group O. So there is no strange and there is nothing weird about these results. Moving to the case of incomplete dominance, which is called also the intermediate dominance. So here we have in the snapdragon plants, a cross is performed between 2 breeding lines, one having red flowers and the other having white flowers. In these cases, the alleles are designated by capital letters, as well as the case of codominance. Both of them, they are designated by capital letters. So here, let's make this example. The parents have red flowers is crossed with another parent having white flower. The result is we have pink flowers 100% in the F1 generation. Okay, so here red and white, we didn't get red in the F1, or we didn't get white in the F1, we get pink, which is intermediate, which is in the middle between the red and the white. So it's called intermediate dominance, where the parents, they are going to be pure, and the both allele, they are dominant. So here the parents, they are pure, so let R capital be the symbol of the red allele, red color allele, and the W capital. So both of them, they are capitalized, so it will be RR crossed with WW. And let's move now to the results in the next slide. White, after crossing F1 with F1, what do we call crossing F1 crossed with F1? Is called the selfie cross. So crossed pink with the pink, so we get 25% red, 50% pink, and 25% flowers having white color. So here let's start with the factorial analysis. So let's start with the phenotype of the parents. So phenotype, male and female crossed with the female. Here note that you have to give the symbols before you start. So you have to say let R capital be the symbol of the allele coding for the red color, and let W capital be the symbol of the allele coding for the white color, which is in the case of co-dominance. No, this is the case of intermediate dominance. So male and female, it doesn't matter since they didn't give us the information about the male and the female. So let's start with the red, and here we have the white. Moving to the genotype. So genotype of the parents, here we have pure RR crossed with WW, the female, and then we move to the gametes, symbol gamma. Here we have 100% R, and here we have 100% W. Moving to the table of cross. So table of cross will show the following. So here we have the table of cross divided into two, male and female. So the male R 100% and W 100%. So they will be RW 100%. So table of result will show that. Table of result will show that we have the genotype will code for the phenotype. So genotype and phenotype. So the genotype, it's RW 100%. So 100% of the individuals will have pink color. Of the genotype R capital, W capital. How can we differentiate between the case of co-dominance or intermediate dominance? It depends on the question and the given in the question. Let's move to the result after crossing the F1. Here the F1, all of them, they have pink color. Of genotype R capital, W capital. Let's to the second cross. Upon crossing F1 crossed with F1, what do we call it? By the selfie cross. So F1 crossed with F1. Let's start first with the phenotype of the parents. So phenotype, male and female. So phenotype, they have pink crossed with pink. Okay, so here we have the genotype. So the genotype, we have in the male, we have R capital, W capital crossed with R capital, W capital, and the gametes will be as follow. Gametes, symbol gamma for sure. So gametes will be R 50% and W 50%, while here it can be R 50% and W, which is 50%. Okay, so now let's do the table of cross. Table of cross, and finally we'll move to the table of result. So table of cross, we have to draw the table of cross, divided into male and female. So first divided male, female, R 50%, W capital 50%, R it's 50% also, and W capital it's 50%. So now let's move to the genotype of the descendants, the probability or the percentages. So R cross with R, so R it's going to be RR, and what? It's going to be 25%, 50 times 50 over 100, and RW, so it will be RW 25%. Also here we'll have RW 25%, and the final will be WW 25%. The table of result will give table of results, so it will be like this. The genotype will code for the phenotype. Okay, so here we have RR 25%. We don't have other than this as RR 25%, so this will give 25% having red color, so red flowers. Then we have RW, it's going to be 50%. How? 50%, here we have 25, this one, and here we have 25, so 25 plus 25 it's 50, so 50% having pink color, so 50% pink flowers. And the last one, which is WW, so WW 25%, so 25% white flowers. Since they gave us the results, and this question mainly they ask you to perform the essential factorial analysis to verify the experimental results, or the results that are given, they are 25% red, so here we have 25% red, 50% pink, we have 50% pink, 25% white, 25% white. So here it's the results, they are verified, so you have to write finally, therefore the results are verified. Okay, here mostly we are working with the percentages. If I want to work with proportions, it differs. So the 50% will be 1 over 2, 25% will be 1 over 4, and the 100% will be 1 over 1, so you can write it as 1. So here, if I want to work with proportion, so you have to be careful about these notes. Okay, thank you guys.